Hi everyone and welcome. Today I want to tell you about our work on direct translations between MRI and histology in the human brain. If you compare it to what Google does, our work is actually not that different. What Google does is it combines images of the Earth created by a whole bunch of different modalities, bringing them together in a single space, annotating the data, creating maps and atlases that you can reuse, for instance, for planning your trip to a conference. What we do is we create images of the human brain using different modalities, we annotate them, and we derive maps which you can reuse, for instance, for your fMRI analyses. So we started this a whole bunch of years ago already, and the first modality we started to investigate and acquire was in vivo MRI. And we acquired a big data set ahead, which is available for download if you like. And what it contains is 7 Tesla sub-millimeter whole brain data sets. They are intrinsically aligned contrasts, because we acquire them using a single scan acquisition, and we have images of 105 volunteers. We cover the entire adult lifespan, so inclusions were between 18 and 80 years old. The data are freely available, and more of it is actually coming, because at the time that we acquired the whole brain data, we also decided to acquire a partial coverage scan that allows us to visualize the subcortical areas of the brain with more detail. So those data are available at 0.5 millimeter isotropic. So what do we use these data for? We use them for our imaging purposes. And how do we do this? We have a subset in this database of 10 individuals. And what we do is we manually annotate subcortical structures in these individuals. We do that twice, so we can create an inter-rater reliability calculation. We use the dice coefficient and the dilated dice for that. And then afterwards, we funnel all of this information in an algorithm called MASP. And what MASP does, it learns how to detect and delineate these structures, and we roll it then out on all of the other subjects in the database so that we create individual delineations of all the structures that are in MASP for all the individuals in the database. What can we do with those? Well, we can model the MRI parameters, but we can also model shape, we can model volumes, we can model the position of these structures across the adult lifespan. So you can start investigating iron accumulation or volume changes, or the shifts that many structures show towards a more lateral position as people age. So this is what we do with the in vivo data. And we have released 17 structures so far, but of course we are still working on a lot more. So these are the structures that we are working on right now. It's a lot of work, and we know we're nowhere near done, but it's a good start. Now, of course, we cannot see all of the structures that we know are in the brain on the in vivo MRI. So, unlike the cartographers from the past who just drew in dragons and said, well, that must be where the dragons live, we decided to opt for a mixed methodology. So we wanted to combine post-mortem MRI with histology, because histology will give us the anatomical detail we need to get all the smaller structures of the subcortex visualized as well. But of course, if we want to reuse this information in vivo, we have to be able to translate it to MRI data. So this is what we did in our approach. So this is then first the MRI that we acquired. This was done by Nick Weiskopf and Evgenia Kirillina at the Max Planck in Leipzig. And you can appreciate the already crisp detail that you get in the thalamus. Now what we do after we perform the MRI scanning is we slice the brain. And this is what you see here at the top. This is our system. We set the machine in motion. It starts slicing, and you can see a 200 micrometer section roll off the tissue block. We capture that, and all of these individual sections, there's 800 to 900 per brain, are stored in small jars until we can further process them for microscopy. Now what you see running in the bottom here is the block phase imaging. So every time we section, we take a picture of the tissue block that is left. And if we recreate all of these photos in a stop motion movie, this is what you get. You get a full reconstruction of the brain already. This is a restacking, not a registration. It's not needed. So this information from these block phase images we can already use to create surface reconstructions. But we can also create a more detailed picture already of the internal structure of the brain. So this is the first step we do. But then we still have all the sections that we collected in all of these jars, and we roll them back out and we mount them on glass slices and we perform stainings. Here in B you see a hematoxylin and eosine staining, which we use for basic neuropathological assessments, and at the bottom you see a calcium binding protein, which we use for getting more anatomical detail in the individual structures. So what we did then is we brought everything together in a single space, and we now have the MRI, we have the block phase, and we have the individual stainings. And all of them, you can switch between them. You can open them at the same time, you can overlay them, you can go back and forth. And we created a full reconstruction of a single brain, which we then offered to a journal. But unfortunately, the journal people said, the reviewers said, well, this is not enough. This is only one brain. So we did it again, and we also released the second brain. Now, what can we use this type of information for? I am, of course, interested in mapping the subcortex further, and this is just an illustration of the potential of the contrast that you get from it. So here again, you see an image of the thalamus, and you can start discerning the individual nuclei that are present in that thalamus. Not everybody is trained in the anatomy of the thalamus, so some of you may be shocked to hear that the thalamus actually consists of approximately 30 individual nuclei. And we won't get at those using in vivo MRI. So we can use the postmortem data here. Of course, the postmortem data also has other potential, and if you want to study cortex, you can create cortical inflations, as you see here. And Pilou has also been playing around with mapping the vasculature. So it does give you a lot of potential, but it's not all positive when it comes to the postmortem work. Sometimes you run into stuff that you rather would not. And this is something I want to give you an example of. This is the original group of donors that we had available for our research. Some of them went through the pipelines completely, some of them did not. I want to draw your attention to donor number 5, a 91-year-old female, which was registered as a non-demented control. We processed the brain as such, and what we found was that there were no gross abnormalities, which we observed on the MRI, and the shape of the brain fit with the advanced age of the donor. Now, what we did then was a routine assessment of the neuropathological alterations that could be visible on the H&E staining. Now, luckily we did, because our board-certified pathologist told us that this was not the brain of a non-demented control. So what you see here is a full reconstruction of the amyloid plaques in red and the neurofibrillary tangles that are presented in green. You can see that the amyloid plaques are largely distributed over the cortical areas, whereas the neurofibrillary tangles are confined more to the entorhinal cortex as well as the hippocampus. So we can conclude now that the postmortem data provides us with unmatched anatomical detail, which we can translate to MRI space. It does also show that we need to carefully check all the neuropathological statuses of all the donors that we have available. So with that, I would like to tell you that we're planning to extend MASP using both the in vivo as well as the postmortem data. We're gonna release additional databases, so keep an eye out. We'll be releasing the 0.5 millimeter partial brain coverage data we have that we created in vivo. We plan to share the Alzheimer brain data. We're gonna work on annotating the postmortem data as well, and we hope to bring out atlases on these at some point as well. And what we're working on right now is to process additional donor brains with Alzheimer's disease. Now, of course, this has been a major project with a lot of people involved, and I would like to thank everybody presented on this slide. And I hope that the chat will work so I can answer your questions. Thank you for your attention.